Assalamu alaikum. Today our lab is about diagnosis of fungal infection. The steps of the diagnosis of fungal infection. We should know first of all the appropriate methods of collecting the specimens, which is very important for accurate diagnosis. The types of specimens depends of course on the site of the infection. If we have superficial infection in the skin, hair or nail, or if we have a systemic deep infection, so the specimen depends on the site of the infection. First of all, the hair. We should pull the affected hair and transport it in a sterile petri dish, cut into pieces, some for inoculation and other for direct microscopical examination. Number 2, skin scraping. The scraping of the skin usually should be done from the outer edges or from the boundaries of the lesion, not from the center, because usually the center is the healing area. The microorganism usually concentrated on the outer edges or on the boundaries of the lesion. After, of course, sterilization of the skin with 70% alcohol. Number 3, nail scraping or cutting, the same. It should be processed just like the skin. In this picture, these two pictures show the process of scraping of the skin by a special knife after sterilization of skin with alcohol. This picture showing the scraping of the nail. Of course, depending on the site, if we have superficial onychomycosis, distal or proximal, so according to the site of the infection in the nail. Number 4, or the fourth specimen, we have the blood and the bone marrow. These two specimens should be centrifuged together with the CSF. Number 5 also should be centrifuged for culture or for direct microscopical examination. Number 6, the abscess, or if we have abscess or wound exudates, we should do aspiration from the abscess and from the wound exudate for examination. Exudate granules or grains are crushed into the culture media and examined directly for subcutaneous mycosis, for diagnosis of subcutaneous mycosis. Number 7, if we have respiratory or pulmonary mycosis, our specimen will be the sputum or transbronchial aspirates. Direct microscopical examination. If the specimen is taken from a keratinized part like skin, hair or nail, we should use 10 to 20% KOH. So we put the scraping of the skin or the nail or the piece of the hair on the slide, and then we put a drop of a KOH with a slight heating. The KOH and the heating will help in a breakdown the keratinized tissue and help to visualize the fungal elements. Other method for microscopic examination is by using the KOH plus chalcofluor. Chalcofluor is a fluorescent dye. Usually, this slide should be examined by the fluorescent microscope. Number 3 is the Indian ink. Also, this stain used for the examination or the demonstration of the capsule for Cryptococcus neoformans in CNS infection. Number 4, tissue stain by using periodic acid sieve and Gims stain. These pictures showing the slice which is prepared usually by the direct KOH and the KOH plus chalcofluor or by Indian ink. For isolation of fungus, we use usually a culture media, which is the Saboroid dextrose agar. This media used for isolation of the fungi. We should add to this media antibiotics like cyclohexamide and chloramphenicol in order to inhibit the growth of the bacteria. Number 2, or the second media used for the isolation of fungus, is brain heart infusion. For the incubation, the molds usually should be incubated at room temperature at 25 to 30 degrees. For the yeast, it should be incubated just like bacteria at 37 degrees. The identification of fungi usually depends on the examination, on the microscopical examination of the culture, on the colony morphology, and of course on the microscopical examination. For the microscopical examination from the culture, if we are going to prepare a slide from a culture media, so we have many methods. The main direct mount. We have a main direct mount by using lactophenol cotton blue stain. This method includes take a part of the colony and teasing it over a slide, put a drop of lactophenol cotton blue, and then put a cover slip and examine under the microscope. The second part is, or the second method is by using a cellophane tape. This cellophane tape is put directly on the surface of the culture medium, and this tape is put on the slide over a drop of lactophenol cotton blue for examination. The third method is the slide culture technique we are going to discuss now. The slide culture technique, or the slide culture method for preparing a slide from a culture media. First step, we should take a sterile petri dish. Then put inside this dish 2 glass rods. Over the glass rods we put a slide, sterile slide, and then we take a block of non-inoculated saboroid dextrose agar, about 1 cubic centimeter. Put this block over the slide, and then we should inoculate the 4 sides of the block with spores. Then put a cover slip over the agar or over the block. Then incubate. After growth, usually we remove the cover slip, put a lactophenol cotton blue drop on the slide, and then put the cover slip over the slide and examine under the microscope. For the block of the media should be discarded. This technique usually used in order to obtain the complete structure of the fungus. We can see the complete structure of the fungus by this method. For biochemical tests used for identification of fungi, the miscellaneous tests for identification of molds are hair perfusion test, number 2 urease test, number 3 trichophyton agar. Miscellaneous tests for identification of yeast, we have the germ tube, carbohydrate assimilation test, or the API 20, corn mean agar for development of plastoconidia and the chlamydoconidia. All these tests we are going to talk about in details in the next lab. Today we are going to discuss the molds, and we are going to take the first class, which is zygomycete. We will have an example for this class, which is genus Rhizopus. For the microscopical appearance, this slide shows the microscopical appearance of genus Rhizopus. The hyphae are aseptated and broad with stolons. The stolons are horizontal collections between the organisms. At the point of the connection, clusters of rhizoids, as you see in this picture, clusters of rhizoid, which is a root-like hyphae, are attached to the substrate. The spore angiophore is the hyphae which is carrying the spore, which is called spore angiophore. Usually terminate with a dark spherical spore angium. The spores, or the spore angiophore, could be spherical or oval, bluish or black with a smooth wall. On the saboroid dextrose agar, the colonies of Rhizopus grow very fast, filling the plate with a loose white to gray hyphae, becoming dark and black in color after maturation of the spore angium. The second class is Detromycete. We have an example is genus Penicillium, which is regarded as a light fungus or hyaline fungus, which is not pigmented. Microscopical appearance. The hyphae are septated, developing brush-like conidiophore which terminate with flask-shaped fillets. From the tip of the fillets, unbranched chains of conidia arise. Genus Penicillium appear just like a brush under the microscope. For the culture, the colonies grow rapidly with a powdery to velvety texture due to the abundance of conidia. So it gives a powdery texture or a velvety texture, and the color usually is bluish to green. The other genus is Aspergillus, which is also regarded as a light fungus or hyaline fungus. Microscopical appearance of Aspergillus. The hyphae are septated. An unbranched conidiophore arise from a specialized food cell. The conidiophore is enlarged at the tip to form a swollen vesicle. As you see in this picture, there is a swollen vesicle at the tip of the conidiophore. The vesicles are completely or partially covered with flask-shaped fillets, and the fillets produced chains of rounded spores, or called gonidia also. Conidia means spores. Okay? For the cultural characteristic or the colony morphology, on the sapropodextrous agar, the colonies of Aspergillus are slow or rapidly growing, varies in color, could be white with green or yellow shades. Usually it appears yellowish green or yellow to green in color. The other genus is Cladosporium, which is regarded as the dark fungi, diatomaceous fungi. So what is the difference between the light fungi as Aspergillus and Penicillium and the dark fungi like Cladosporium, or how we can make an identification between them? If we look to the undersurface of the culture media, usually the dark fungus will appear dark in color, while the light fungus will appear light in color. There is no pigmentation in the undersurface. For the microscopical appearance of genus Cladosporium, usually the hyphae are septated. The conidiophores are dark, varying in length, branching irregularly towards the tip to form a tree-like conidial structure. So it's very similar to the tree branches. It is just like a tree under the microscope. For the cultural characteristic, the colonies are slow growing. The color is olive green to brown. The reverse side, as I said, or the undersurface of the culture media, is olive green to black powdery in color. يعني the reverse side of the dark fungus is also pigmented, and this will differentiate it from the light fungus, which is light in color from the reverse side. And that is all about our lab. Thank you for listening.